Welcome, everyone. Welcome to our September community meeting. We're really excited to have everyone joining us from all over the globe. Today, we have our speaker representing First Approval. So start by introducing him. So joining us today, we have Timofey Glinin. He's a molecular biologist and a postdoc scholar at the University of California, San Francisco. He is the co-founder of First Approval, a startup building infrastructure to make scientific data publishing open, collaborative, and beneficial for researchers. With more than a decade of experience in genetics, biotech, and aging research, Tim is committed to accelerating science through open data and innovative approaches that improve reproducibility and collaboration. And he'll tell us more about the student biological data competition. And I'll let the room for you, Tim.
Thank you. Thank you. Hi, everyone. Today I will briefly speak about open science in general, and next go to one of potential solutions for students, which is First Approval Student Biological Data Competition. So let's start from the problem of science in general. So what we want to have from science is like basics of science is that experiments should be reproduced and could be reproduced by other researchers. This basic thing is important for like overall efficiency of scientific process. Simultaneously, a lot of problems report that they have problems. A lot of scientists report that they have problems with reproducibility, and more than 90% of them at least sometimes struggle with reproducing some results. One of the reasons why these results could not be reproduced is, and one of potential solution is availability of scientific data. Because right now, when you read the article, you see graphs, which is some kind of interpretation of data. But there are 99% of original raw files is unavailable. And most of the time, processed data is also unavailable. And this threatens the progress of entire field of scientific research and give us an average number of only 30% of experiment could be successfully reproduced from first try. So scientific institutions recognize these problems and start changing the policies with much more open data and open science recommendation. So it is general for all countries. There are UNESCO recommendations, there are specific data sharing policies of NIH, Europe science finding organizations, Chinese state council. So it is like a general situation that right now all institutions try to make data more transparent and simultaneously get much more detailed annotations for this data. Final and in trend of this activity, we try also to put these new practices of data annotation and data publishing available for new generation of scientists, for students. And for final brief overview, what is data set could be and why it is important to publish data. So It could be their data set could be even large or small, for example, and contain different types of files. Sometimes it could be just small Excel table with, for example, from behavioral tests, and sometimes it could be a large specialized files from next generation of sequencing or other like specific devices. It could be images from microscope or raw files from microscope. We will get into this later today. But to understand how much the data sets are missing right now, we have about 2.5 million of scientific articles per year with low replication rate, unfortunately. And behind these articles, it is about 25 million of data sets. Now less than like 1% of them are annotated in detail and available. And we believe that making this data open could also provide additional insights for some kind of reanalysis of this data by independent researchers, because it is not only about solving the reproducibility problem, it is also about making new insights from reanalysis. So from our side, we as firstapproval.io innovative repository and data publishing platform, we decided to run a new initiative, student biological data competition. It is international contest dedicated for scientific data sets in biology, biotechnology, and biomedicine. It is first of its kind competition directly evaluates the quality of scientific data sets and their annotation, aiming to promote open data practices among students. The central focus of the competition is the evaluation of scientific data from experiments. Doesn't matter which specific results could be get after interpretation of this data. So we will focus on data and quality of files and annotation first. The competition is open in three categories for bachelor's, master's, and PhD students. Their datasets will be judged separately in different categories. Submissions will be evaluated by a panel of experts based on completeness of annotation, data accuracy, novelty, and quality of experimental design, as well as potential for data reuse. Participants will have opportunity to gain recognition from independent panel of top academic and industry experts in their fields and will receive data publication on first approval platform. So submission deadline is 15th of October 2025, but we have rolling submissions. So if you will submit your data set earlier, it is bigger chance that it will be processed in more like accurate and details, because to the deadline we probably will have a lot of submissions. But another information that right now we have not too much submissions, and it is high chance that you as participant, student, could win our prize money awards. The first prize in each category is $1,000 and the total prize fund is 7.5 thousand, which will be distributed for 32 winners. And we have a special prize for best negative dataset and best replicative dataset where experiment was successfully replicated. Also, all participants who reach the final selection stage will receive a certificate of honorable mentions, and winners will be announced on our website and ceremony. After the competition concludes, all participants who submit their dataset and overcome quality check will have their dataset published with open access publication on first approval platform with available PDF version of their publication. Executive jury board right now is 5 professors from top universities from United States, UK, Europe, and Asia, including Albert Einstein College of Medicine in New York, George Mason, and others. We have an organizing team which led by Daniel Dominguez. I also part of organizing committee. And if you will and wish to share information about competition in your university and you're ready to help us with processing dataset and you are a scientist in some scientific institution, we will be, or like industry scientists, we will be happy to invite you also in organizing committee. So if so, please contact me. Some leading universities is already support and push forward their students and communities to publish and submit datasets for competition. For example, University of Cambridge recommends to participate in competition, and biggest open data initiative, Research Data Alliance, also announce our competition on their website. So why to join for if you are a student, small summary, you get your first, or probably not first, but publication. Your dataset will have DOI and accounted as real scientific publication that you can list in your CV. Get real scientific focused skills, learn how to organize and annotate data way real scientists do, and write scientific publication. Stand out with, if you win the competition, you will get awards and diplomas, money prize, and show to your future career opportunities, increase your future career opportunities. And you can tell that you participate in initiative in open science. Additionally, connect with mentors. You can work together with your PI and get credit as co-author. So you get this kind of experience to processing your publication to submission, and make your project matter. Even a student experiment can become contribution other scientists will use. So today, the next section will focus on how to get real scientific focused skills, and I will specifically go through submission process of our competition and give some tips about how to make this submission successful and get chance to win the prize. First of all, submission could be done at firstapproval.io/contest website. Here is the QR code, and if you have any questions, you can write for our email competition@firstapproval.io. Next, you will come going through this link to competition webpage. Here is the guidelines, so you can download them or push apply now button to directly come to registration process. We will registration is simple and standard. You can register by email. Also option with ORCID registration. You will need to provide your name and institution affiliation, and then you can go for processing of your submission. So you if you already registered, you can just push the apply now button and choose the data collection, student data competition. It will be just 3 options, and for participating in competition, you need to choose this one. And the submission, like general submission page, will look like this. So information about your academic level, title of your dataset publication, research area, summary, background and aims, materials and methods, data description, optional section of preliminary result, optional section is software, files and signed letter from your academic supervisor with template of this letter. If you have granting organization and literature, like you can put it in this section, and related publication is literature which you used during making this submission. Final is tags for make your dataset findable. Yeah, in brief, you probably would like to start with background and aims section where describe where you describe goals of your experiment, material and method sections where you describe processing and experimental design, and the data description is which files you uploaded, how they organized, and all specific things about your dataset files. And software is a section where you potentially can describe which kind of software you use to process the data, and which software could be used for reanalysis of your data. So yeah. But now I will come to tips, specific tips, how to write this section of dataset annotation. You can start by reviewing method sections of your lab's previous publication. Additionally, you can search high quality literature for how similar experiments are designed. Revisit your own lab notes to capture every procedural step, and make a checklist for critical details, reagent name, concentration, supplies, equipment, model, if some serial or specific software program details, you also can put it in dataset annotation. Any calibration details, software name, versions, and key settings. So it is something that you can directly, just if you find some information for this, you can put it in your dataset annotation. In general, you can ask yourself just simple question: would another researcher be able to repeat this experiment using my dataset annotation? If the answer is yes, probably this means that you made a good data annotation. Specific tips about writing the experimental description in sections of background and aims, design flow, and technical documentation. For background and aims, you can cite prior similar experiments that related to your study. Highlight key findings that shaped your research question, and state your goal of your experiment clearly in a single standalone sentence. Design of experiment and design flow could be described by you just need to ensure experimental design logically follows from aims, report sample size and object characteristics, and describe preparatory steps, experimental process, and data processing. So, technical documentation, just lists of software versions, parameters, and file compatibility details, describe folder structure and file naming, which you used, indicate how raw, processed, and analyzed files are organized. So it is just a brief demonstration how it could be done on first approval platform. It is like old video, and not specifically for this competition. You in general can upload any kind of supplementary data in first approval using the first approval general data collection. So it just describe how the process of file uploading, author, how you can add co-author, write literature, text, and all these steps. After you finish annotation process, you will come to next step of finalizing submission. We will discuss this too. So, in this competition, research areas. So when you will push it, it will have like this list of research areas, including like standard biological areas like cell biology and histology, immunology, neuroscience, developmental biology. We have specific section omics technology. So if you would like to submit some kind of large data of transcriptomics, genomics, metabolomics, or anything else, you can put also omics technology section notification. But if you would like to submit small simple data set of like some kind of Western blots, PCR electrophoresis, anything like this, it also could be valuable data. So we will accept this kind of submissions too, and they also could win some prizes depending on jury decision. In background and aims, you additionally will have three check mark, if your data set is negative, you should put this check mark, turn on this check mark, and you will be invited to write why you think that your experiment not support initial hypothesis. If you have successfully replication of previous experiment which was already published, you can check mark on this section and write description of previous experiment that was replicated by your experiment. And additionally, we allow to submit previously published data sets, and if you will increase the data annotation. So if you describe your data in much more details, you can even submit your data set to competition and write here your previous data publication and what the improvements were done. So, but if you your data set is not negative, not replicative, and haven't been previously published, just leave this field like this and describe the content, like general background and aims content here. So in this section. Next, specifics about file uploading. So you can drag and drop files to this field, or either you can push the upload button and navigate through your computer to find single or grouped files. Additionally, you can make folders to organize your data if you would like to. For example, you can make two folders for raw and processed data, which would be better to upload both if you like have this type of raw data and processed data. And additionally, you can make small notes for any type of file that you uploaded here. So in addition to name, you can, like, for example, if something specific with single file which you would like to mention, for example, this particular run of experiment was longer than others for like some time, you can indicate it in this section that this particular file was processed longer, for example, or any kind of other annotation details, what you think is important for like potential analysis of data and data reuse. So data, some tips about data in file documentation. Core data like main files should reflect the full experimental flow. You can add supplement with photographs with captions which you make during your experiment procedure. You can upload any supporting data, include quality control results, for example, nanodrop readings, dilution tests. If you have like some pilot results that you like run before original experiment and they are relevant for this data set, you can add them too. If you have complex design experiment like multi-steps workflow, you can attach paint and attach experimental schema as an image if you think that it will make like your procedural steps clearly. on this schema, you can represent the flow, timing, and case steps, for example, if you would like to include this. Annotation practices like label every file clear with experimental data, condition, use notes for files if necessary, and keep supplementary material linked to main dataset for clarity. After you will finish your annotation process, you will go to this page where you finalize your submission. You will see the preview of like name and summary. Name should be less than 200 characters, summary 300 characters, so it's like short abstract and name. And you can double check them. You can double check the correctness of all data set. So you will have option to download draft PDF. And it is just notification that your publication will be open access, that it will be published under standard license with standard like citation rules, and you can choose in which storage you will we will store your files. You can choose whatever you want. It is just like two different options: cloud storage or decentralized IPFS storage. And next you will have the check boxes that you will need to mark. And next you can proceed for like submit your publication. After the all data sets will be published, it will happen after we will finish all submission process, and after initial evaluation of all data set, we will publish all of them simultaneously. And the publication will look like this. So it will have name, DOI, first approval link, information about the data, like how many folders, files, and the overall weight of these files, and so articles, affiliations, so all fields that you will fill will be represented like this in PDF file, which could be open downloaded in open access format on first approval repository and our web page. If you already have, a lot of questions is raised if I will next publish my dataset in scientific journal article. Is this data publication, participation in competition will somehow disturb this future publication? The answer is it will complements later publication in peer-reviewed journal. So the process will look like this. You participate in competition and publish data at first approval platform. And then if you simultaneously prepare the scientific article, you will put this data set in data availability section of your journal publication. So in data availability, you just this section exists in any kind of peer-reviewed journals, and it's specifically designed to notify where your files are uploaded. So you just need to indicate here that files from this experiment is uploaded in first approval repository and provide a link and DOI. Interesting fact that integration of data publication into journal article will increase the citation score of your journal publication. There are like confirmed statistics from open science research which indicate that citation score of articles with available data is higher than without data disclosure. So if you already have journal publication ready, you can, but your files haven't been published yet before, you can publish these files in this competition. You allowed to publish these files and annotate them. So you just need to cite article where you previously published your publication. So it's not disturb any kind of process related to peer-reviewed journal publication. If you have your publication already done, but you haven't annotated data, you just upload your dataset. If you plan to publish journal article, you just integrate your information about your dataset in data availability section. So we have specific section for types of specific data types. I will briefly go through it because it just will like give some tips about standard experiments that students regularly run. And just to like get to any participant understanding how it could look like. So most standard technique in every student laboratory is PCR plus gel electrophoresis. And you can upload gel images in TIFF and or JPEG files. So raw data will be unlabeled, processed data will be labeled images. Just some tips about annotation, record agarose gel percentage, buffers which you used, note DNA letter type and band size, ensure letter and visible letter is visible in raw image, provide uncropped gel images with all lanes, samples control markers, label lane identities, like which are control and which are sample, and design the whole experiment is also need to be provided. It is just tip for this, how to annotate this specific data type. Like for qPCR, raw files would be like instrument raw files or RDML files. The processed data will be CSV or Excel tables. So, and annotation tips will include primer sequencing, target gene ID, amplification length, probe fluorophores, document all PCR run conditions and reagent details, indicate reference genes that you used or any calibrators or standard curves used for quantification, ensure technical and biological replications are identified, and include threshold settings or baseline correction applied. For microscope files, for example, so both light and microscope are available, but for microscope files, fluorescent microscope. Different microscopes regularly have their own raw file standard. It could be CZI file for Zeiss microscope or other files for Leica and Nikon and other microscopes. The processed files could be exported TIFF files with scale bars, with they could be single channel and multi-channel to make it more like consistent. For annotation tip, record microscope settings, instrument, objective, illumination, filters, lasers, detector. Document all image processing steps, for example, deconvolution or contrast adjustment. Use consistent file naming and folder structure, like experiment, date, stain, replicate, and use nodes if something specific with your files. And the final like example that I would like to notify you is behavioral test. Like, for example, if you run some behavioral test in student groups, you can the raw files would be individual participant data records. It could be spreadsheet in Excel files, for example. And processed data could be summarized tales about every participant. You need to make sure that everyone is anonymized, and you put only anonymized participant metadata like age, gender, group assignments. Share raw response per participant in structured format. Define all variables and codes if needed. And specifically, if you have any like any human kind data, remove all personal identifiers and note consent and ethical approval. Like well annotated behavioral data is highly enable for reanalysis and meta-analysis. Like summary of this section, general tips: annotate early and data early and continuously. If you done this in advance, which is regularly like just good laboratory standard practice, you will process the submission much easier. Adopt standard metadata and formats. More details better than less. Check all questions with your co-authors and principal investigators. Organize and structure your data clearly. Provide clear description for all data fields. Validate and quality check your data and annotation before submission. So after you finish all your description steps, you probably would like to double check everything, read it one more time, and correct any potential mistakes. Keep reusability in mind. Data need to be findable, accessible, interoperable, and reusable. It is basic fair principles for data sharing. Additionally, I just would like to tell that the submission process, if you have your like documentation prepared well, could be just several hours for single submission. So you can just try to start doing this, and maybe you will submit your application fast. But sometimes if you will like struggle with some information, it could be difficult to like find exact information, like some detail that you don't know where to find it in previous records. In that particular case, we recommend you to start submission process in advance, so you will be prepared if you will need to spend some time for this check of details. And the last thing, that it is open science initiative. We created to make like the data much more open, to make scientific standards much more like clear for everyone. And we believe that this competition could become like really like great experience for every student. And we like need support in raising awareness about this competition. So if you can tell about this competition to students around you, it will be really helpful. We have media kit on our website with printable flyers, which you can distribute across your institution. So if you don't see this flyer on walls of your institution, you definitely can print and put it there, because like we would like right now is active step of this competition, and it's enough time to process any kind of submission. So yeah, it's available on our website. Ask to share information, your student organization, ask your department probably to share information through email list. And yeah, media kit is available on our competition website. And the final is link for the competition, and I will be happy to answer any your questions. Thank you. Thank you for being here.
Amazing. Thank you so much for sharing and bringing this opportunity to our community call today. It is really exciting. And I mean, I hope that everyone here as a good scientist is also excited to share their negatives also. So sharing also what didn't work. And exactly, open science is about fair data and making sure it's reproducible and it's collaborative. And this competition is exactly around that. So thank you so much for sharing about the opportunity. I want to again invite everyone here to share with your communities, to participate in this competition. Is an opportunity to share your undergrad thesis, your master's, your PhD, and yes, hope we have everyone, especially today that we have a lot of Latin America representation on the call. So that's very exciting. And now we can open for questions. So Yankee also share first approvals email if you have any more specific questions. But now we have some time during this call. So if you want to open your microphone or send your questions on the chat.
So I can kick off with one whilst folks are thinking about all the exciting data that they could be uploading. So you said that this is open access as well. So would people be able to look at the data set without signing up to first approval? And equally, can people then help build on the data that's been provided?
Yeah, so we try to create. So what we found in first approval, that regularly all data authors would like to know who download the data, and we would like to implement some kind of new standard of open access. So we don't make any borders for registration, but we make publicly available the information who particularly download this data set after the registration. So we believe that it could be much more transparent and clear sharing data with this kind of improvement, because it could be the first step of potential collaboration, or because if you know who exactly are interested in your data file, it could like make you want to communicate with this person, or some kind of interact, like check what articles this person published. So we believe that from that perspective, it is much more beneficial not to put downloading borders, but just make this downloading notified and tracked who were interested in this kind of data set. So from that perspective, we allow to download data freely, but after registration. Yeah.
No, that makes sense to be able to try and encourage more interactions between
Yes.
submitters and people interested in the data.
Yeah, definitely. I mean, this has been something that we also discussed a lot on Recon with the collections. We share, they are open for you to share with others, but at the same time, it's good for us exactly to keep track of, what labs are we reaching, and even the troubleshooting side, and like, oh, this worked in a lab, this didn't work in another. And yes, I think it's a very interesting system that we use. We use our forum for Recon also, where we share the community calls to have, exactly this open space for discussion and knowing where our work is getting.
Yeah.
Amazing. Do we have yes.
Just brief notion that regular data publications are not downloaded too much. So there are some kind of really impactful data sets that is downloaded thousands of times, but regularly the small amount of data do not have too much persons who are interested in. And from that perspective, we think that the value of these persons could be increased for the author if it is possible to know who is this person who are interested in your data. So yeah.
Perfect. Do we have any other questions, comments?
Hi, Sibylle, can you hear me?
Yes.
Okay,
Hello.
Timofey, I don't really have so much of a question as just a comment that I think this is great what you're doing. This is the first that I've heard of this contest, and I've already been telling multiple students I know about this. So I congratulations on doing this. I guess I think maybe I do have one small question about the data sets that you asked for. Is there any sections that might include, for example, if students are using predictive AI for things like AlphaFold, for that, would that qualify for this, or would you prefer that it's something more physical, right, per se?
So it sometimes could be the question. We would like to have any kind of raw or processed files rather than just analysis. Yeah, if you can provide some kind of data set that you generated somehow, you can submit it and describe the process, how you generate it and why it is important. But if it is just an analysis of data, so you get access to some kind of database, you go through it, or you improve something, or you find, it's probably not the focus of this particular competition, because we would like to evaluate data, like any kind of data specifically. Yeah, so it is.
Yeah, okay, that's great. I have students who've done things like AlphaFold, but I also have students that are doing 16S PCR and sequencing. So that's generating chromatograms, generating raw data. So
Yeah.
but great job. Thank you for presenting today.
Thank you.
Perfect. Do you have any other questions?
Not particularly a question, but also I just really appreciated how you had that little checkbox for negative results as well, because I think this is not something I've heard of before being presented, and it's good to see, what didn't work so people can actually build on that as well. Yeah, overall, great idea, great concept. And looking forward to sharing this with our recon community and wider communities to see getting more students to sign up for the competition.
Thank you.
Thank you.
Yes,
And
definitely.
actually, as a brief call to action, it would be great to have more people, more students submitting negative results. I think that could be really powerful and impactful for science. So if you could also make this a specific invitation to students, like incentivizing, empowering to share this, I think it would be great, and we would really appreciate it.
Of
Yes.
course, it's a great learning curve, so we'll definitely do that and call out to everyone on here to share this to at least one other person then.
Perfect. Yes. I mean, maybe we can wrap up exactly with what Yankee said. So everyone, I know all of you here have data that you could share. So here's a great opportunity, especially the negative results, so we can continue building a more collaborative and open science for all over the world. I don't know, Tim, Danielle, if you want to do some last comments.
So thank you everyone for participating, and if you interested to, like, if you some kind of interested to become organizing member, if you support this initiative and you know how to share this information across your scientific circles, or specifically if you know how to spread this information through some kind of scientific organizations, universities, or stuff like that, we will be happy to get your experience in our organizing committee, because it is still challenging to get directly to universities, just to ask them to share this information. And every support in this perspective will be really helpful. So yeah, we'll stay in touch. We will be happy to share any additional updates of the competition with you. And yeah, let's make science open, transparent, and efficient. Yeah.
Perfect. Thank you. So thank you everyone for joining our September community call. Hope to see you in October 22nd. We'll soon post details on our forum. And on our forum, you can also have follow-up discussions on the community call today and all the others. We are posting all the videos, so make sure to get your updates there. Thank you, everyone. Have a great rest of your weeks.
Thank you.
Thank you. Take care. Bye.
Goodbye, everyone. Ciao.